Very good morning to all of you. So I'm Dr. Hardeep Singh Tuli from Maharishi Markandeshwar University, Ambala, Haryana, India. So today I will present my topic on the mechanism of action of natural metabolites in cancer, recent trends and advancements. So as you know, cancer is uncontrolled growth of a cell, and despite the availability of a variety of therapeutic agents, still the cure of cancer need to define. Right, so it is very essential to understand the mechanism of action of natural metabolites because such mechanistic insight will not only provide us the way to treat a tumor, and but it will help us to understand the biology of tumor. Right. So here in this lecture we will study some of the natural metabolites that can be used to that are being used in cancer research very widely. So very first is a cordycepin. So this cordycepin is a very promising molecule, and it is very widely being used in cancer research. So this is the structure of cordycepin. It's if you can see here, it's a nucleoside analog, and its structure is very much similar with adenosine. The only difference is that this cordycepin it doesn't have a oxygen atom here, right? So it's a 3-deoxyadenosine cordycepin. So very promising molecule. It is being extracted from mushroom Cordyceps militaris, and a very promising molecule in terms of cancer research in present scenario, cordycepin. So another molecule is xanthomimol. So xanthomimol, another molecule that is being used in cancer research nowadays. So this is the structure of xanthomimol, right? So it is again a kind of flavonoid. The degulin is there, very promising molecule, natural molecule that is having very important role in cancer research. So this is a chemical structure of degulin. There is a silibinin is there, and again very promising flavonoid, and very well known in cancer research, silibinin. So this is the chemical structure of silibinin. So again another one is the cyclin, very promising molecule in terms of cancer research. A natural molecule, belongs to flavonoid. So this is the structure of cyclin. There is epigallocatechin gallate, and a very promising molecule EGCG. So this molecule is very promising and being used in cancer treatment and cancer research. This is the structure of this EGCG, epigallocatechin. So now again another one is the carcinol, and this is a very promising molecule and being used in cancer research right in present scenario. So this is the structure of carcinol, and it is some sort of chalcone moiety is there. Another molecule is a hesperetin, and we published this report in Experimental Biology and Medicine Journal, and this molecule again belongs to flavonoid, and this is the structure of this molecule, a kind of polyphenolic flavonoid. Another one is a genistein, and very promising molecule in terms of cancer research, and natural in its origin. Genistein, this is the chemical structure. Another couple of promising molecules are quercetin and quercetin, and these are the chemical structure of quercetin and quercetin, and these are also from flavonoid family and polyphenolic compounds. So another, beside these, another set natural molecules or metabolites are curcumin, polyphenolic compound, then apigenin, again from flavonoid family, kaempferol, again from flavonoid family, oleanolic acid, ursolic acid, cholesterol, carnosol. These are from terpenoid family and very promising molecule in terms of cancer research. And isothiocyanates. These are from cruciferous vegetables, and as name suggests, they contain sulfur. Gabogic acid from xanthanoid family. Tocotrienol, kind of this is vitamin E. Then there is melatonin, a kind of methoxyindole compound. So these are various natural metabolites that are very widely studied in terms of cancer research in present scenario. And this is our recent book that we published about drug targets in cellular processes of cancer. Various drug targets we covered in this book. So let's talk about one by one, like the induction of apoptosis. So when we talk about apoptosis is programmed cell death, and here we can say apoptosis is mainly governed by intrinsic pathway and extrinsic pathway. When we talk about intrinsic pathway of apoptotic cell death, it's mediated via cytochrome C and activation of caspase 9, cytochrome C, and apoptotic protease activating factor. This complex further leads to the activation of caspase 3, 6, 7 and lead to the apoptotic cell death, right? So here, this is the intrinsic pathway of apoptotic cell death, right? So when we talk about extrinsic pathway of apoptotic cell death, it generally starts from death receptors like TNF, trail, and Phase receptor, and lead to the activation of this complex, and then further lead to the activation of pro-caspase to caspase 8, and then activation of TBID, and finally this further leads to activation of cytochrome C, and again the activation of this complex, right? So which further lead to the apoptotic cell death or the programmed cell death, right? So here you can see this down arrow is showing down regulation, and this up arrow is showing up regulation, and these stars are showing the natural metabolites. So these natural metabolites, they are known to inhibit or downregulate the anti-apoptotic molecule like BCL2. So these are the anti-apoptotic proteins that are downregulated by natural metabolites, and these natural metabolites are known to upregulate the apoptotic molecules like cytochrome C, caspase 9, caspase 3, 6, 7. Similarly here, caspase 8. So these are the various points or the targets where our natural metabolites can activate apoptotic cell death. So apoptotic cell death is programmed cell death, right? So this is one pathway. So another one is the induction of autophagy. When we talk about autophagy is self-eating of cell, right? So when we talk about autophagy, it generally starts from stress condition, right? So whenever cell feels stress or starvation, then this autophagy cell death pathway it can activate, right, in cancer. So here is you can see during stress condition, Bacillin of VPS34, ATG34, and LC3 molecules, they are get activated, and this activated molecule they further activate the formation of autophagosome, right? From the precursor, autophagic precursor to phagophore to autophagosome, and further this autophagosome they combine with lysosome and lead to the formation of autophagolysosome, and finally it starts self-eating of the cell. So nowadays autophagy is very promising in cancer research. So these are the various points or the targets where our natural metabolites can upregulate. Like our natural metabolites, they can upregulate these molecules so that autophagy can occur in cancer cell. So these are the inhibitory pathway of autophagy, and further these inhibitory pathway of autophagy, they are blocked by the natural metabolites, right? So this is a very promising method in cancer research to cure cancer autophagy induction. So another one is the cell cycle arrest. So when we talk about cell cycle arrest, so cell cycle arrest, very first we should know what do we mean by, or what we mean by this cell cycle. Cell cycle is a sequential changes of cell from one phase to another phase, like G1 phase to S phase to G2 phase to M phase, right? So when we talk about these sequential changes of cell from one phase to another phase, uh, there are some checkpoints that has to cross by the cell, like this G1S checkpoint and this G2M checkpoint, right? And then this M checkpoint, right? So during each and every checkpoint, there is there is a some complex that has to form, right? Like the cyclin D, CDK4 and 6, cyclin E, CDK2 is needed to cross this G1S checkpoint, and cyclin B, CDC2 is needed to cross this checkpoint G2M, right? So what our natural metabolites they do, they downregulate these complex so that a cell cannot cross these phases, right? So here you can see this cyclin D, CDK4, 6 complex is downregulated by the natural metabolites. Similarly, here cyclin E, CDK2 is downregulated by the natural metabolites, so that a cell will not cross these phases, and cell will be arrested either here at the G1S or at G2M checkpoint or M checkpoint, right? So this is another very promising mechanism that is mediated by natural metabolites in order to control the cell cycle growth, right? Or the cell cycle proliferation. Another very promising technology, or another very promising method to inhibit this cancer growth, that is the inhibition of angiogenesis. Angiogenesis is the formation of new blood vessels. So this is a tumor, and this is a developing tumor, and this developing tumor need this new blood vessel, right? Because this new blood vessel formation is essential for the growth of this developing tumor, because via this blood, this tumor will take oxygen as well as nutrients, right? So during this development of tumor, there is a hypoxia condition, and due to this hypoxia condition, HIF1 alpha is highly expressed, and this HIF1 alpha, what it will do, it will activate HF and further lead to the formation of new blood vessel towards the developing tumor. So these are the various pathways that are needed for the activation of HIF1 alpha, right? So what our natural metabolites, what they do, they inhibit these molecules so that HIF1 alpha activity get downregulated. So here you can see PI3-AKT mTOR signaling pathway is inhibited. Similarly, STAT3 pathway is inhibited. Similarly, iNOS pathway is inhibited so as to inhibit the growth, or so as to inhibit or downregulate the activity of HIF1 alpha. Right? So by this, new blood vessel formation will be stopped. Right? So another very promising mechanism that is inhibition of cancer angiogenesis. So another one is the inhibition of inflammation. So when we talk about inflammation, inflammation is generally considered as a kind of mechanism by the body in order to get rid from the diseases, right? Inflammation. So it is a kind of protective mechanism that is adopted by the body during the infection. So inflammation, but prolonged inflammation or chronic inflammation is responsible for the progression of tumor, right? So these are the various pathways or the mechanism by which inflammatory molecules they are activated, like by the activation of STAT, by the activation of NF-kappa beta, right? So these are the various molecules or the factor that are responsible to activate inflammatory cytokines. So these are the various inflammatory cytokines that are activated by these factors like STAT or the NF-kappa beta. So what our natural drug metabolites they do, they inhibit these pathways. They inhibit these molecules upregulation, so as to inhibit the inflammatory microenvironment in of the tumor. So these are the various inflammatory cytokines like inflammatory gamma, right? Interleukins, 1 alpha, 10, right? So these are the various inflammatory cytokines that are down regulated by the natural metabolites, so as to stop the growth of tumor. So another one is the inhibition of cancer metastasis. So when we talk about cancer metastasis or the metastasis is the movement of a cell, cancer from one side to another side, right? So let's say this is a tumor, and if this tumor travel from one side to another side, then it will consider as metastatic tumor, right? So this is the tumor, this is the extracellular matrix. So in order to travel from one side to another side, this tumor has to digest this extracellular matrix, right? So this extracellular matrix has to be digested by the tumor cells so as to travel from one side to another side, right? So in order to digest this extracellular matrix, what tumor has to do, tumor has to upregulate these MMPs or activate these MMP. What are these MMPs? They are the matrix metalloproteases. So these are the matrix metalloproteases. These are enzymes. They are known to digest this extracellular matrix. So once this extracellular matrix is digested, then only this tumor can travel from one side to another side, right? So this is the pathway by which this extracellular matrix is digested, right? So what our natural drugs or natural metabolites, what they do, they inhibit this pathway so as to inhibit the growth of, so as to inhibit the digestion of this extracellular matrix, right? So if the extracellular matrix is not digested, then this tumor will not travel from one side to another side, right? So this is again very promising approach that can be opted by the researcher in order to treat cancer, right? So another one is the inhibition of telomerase. So when we talk about this telomerase enzyme is very essential to maintain telomeric ends of the chromosome. So during each and every cell division, this telomeric ends they get short, right? So, but in cancer, in tumor, they have upregulated telomerase. So this upregulated telomerase enzyme is responsible to maintain the this ends of chromosome. So if the ends of chromosome they are maintained, and then cancer will not die, right? So this high expression of telomerase enzyme, it provides immortalization or proliferation to the tumor growth, right? So what now natural metabolites, they are very promising. They are being very considered very promising in order to down regulate the expression of telomerase. So if the expression of telomerase is get downregulated, then if the expression of this telomerase enzyme is get downregulated, then the growth of tumor, it can be stopped, right? So this immortalization character or proliferative character can be stopped. This inhibition of telomerase enzyme very promising in terms of cancer research nowadays. Another one is the anti-platelet aggregation. So when we talk about anti-platelets aggregation, so that means so this is the tumor cell, and a tumor cell, the migration of tumor from one side to another side via blood. It is here. This diagram is showing the migration of a tumor cell from one side to another side via blood circulation. So here you can see this tumor cell is surrounded by platelets, right? So this is the ability of a tumor cell to be surrounded by the platelets, so as to prevent the action of a natural killer cell, right? So if this platelet aggregation is inhibited, so that means our immune system can identify a tumor cell, and our immune system can destroy tumor cell. But the tumor cell has the ability to aggregate platelets on its surface so as to prevent the action of a natural killer cell, right? So this nowadays some of the natural metabolites, they are being investigated to prevent the aggregation of platelets in the surrounding of tumor cell, like cordycepin. Cordycepin is reported to inhibit the platelet aggregation, so as to prevent is reported to inhibit the platelet aggregation, so that natural killer cell can destroy this tumor cell, right? So here is a summary that what we covered. We studied about cancer. We studied about some therapeutic targets, like we studied about apoptotic cell death of cancer, and we studied about autophagic cell death of cancer, and similarly we studied about cell cycle arrest, and we studied about angiogenesis inhibition in cancer treatment. We studied about metastasis inhibition by the help of these natural metabolites, and we studied about this inhibition of inflammation in cancer or tumor microenvironment. One more aspect is the telomerase enzyme inhibition or the downregulation, so as to treat a tumor or cancer. So thank you very much for listening.